Hello, everybody. Ready to go?
Yes.
Okay. This is your last exam, I suppose, after pathology, and then I think you are all done, correct?
Yes.
Okay, good. As usual, what we'll do is, if you have any questions or difficulties, please ask me, and then we'll go through. I have more than 10 questions, about 10, 11 questions. So you will have a feel of what kind of questions will be coming in the exam. Now, I must say, compared to your last exam, hopefully this is a little less dense unit, and so you should do better than your second unit. Hopefully that happens. It happens all the time. So hopefully this might happen this time. And so some of you who have not done well in your second exam can take advantage of this. I have been answering questions on emails, whoever sends me. Now, there is a different email which you might not want to use because I don't get that, and I have mentioned this in the graduate office. So there might be some emails which I have never received. So in case that has happened, I apologize. But this is something technical, and it doesn't come up on my email if you use a different email. Looks like overall everybody's doing well. Any specific question on anything in any of your chapters? How do you all feel about the signaling and molecular developmental biology part? Is that easy? Just want some feedback from you guys.
It's my favorite material of a lot of the blocks. It seems to be more direct.
Yeah. And we are planning to actually expand a little more because the newer version of the textbook, which has just come out, you're using, I think, edition 10, but edition 11 has come out, and there's more molecular aspects. And increasingly, when you go into regenerative medicine later as a physician or any healthcare worker, you will come across all these signaling pathways and its inhibitors. They're all in either clinical trials or are being used in the clinics now. So that's something which you should keep in mind. So when I say inhibitors of sonic hedgehog, which are used in cancer, certain type of cancers, because sonic hedgehog is highly proliferative. Same is true with Wnt inhibitors. So it could be a frizzled antagonist or directly binding to beta-catenin and things like that. So those inhibitors are also known, and I'm sure you guys know about TGF-beta inhibitors. So the field is growing very fast. In the last 10 years, people have moved from structural and anatomical aspects to more signaling and transcriptional aspects. Looks like if nobody has questions, we can open up the exam quiz. And you can still, ask me questions as you do. Is everybody okay with that?
Sounds good.
Okay. Let me do that then. Okay, is everybody able to see the practice questions?
Yes.
Okay, if somebody wants to volunteer, we can do that one by one, and it will cover all the chapters. So it's not that I have focused on one or two chapters. So that would be better for you guys to brush up. Somebody please start with question one.
A U-shaped D-looping, which converts the cardiac tube into an S-shaped heart, occurs due to which of the following structural changes? Is it C, ventral rightward looping of the heart?
Yes, that is correct. uh, give some time to others. Anyways, who was this? Was it Elizabeth?
Yes, I'm sorry. I should have waited.
No, that's fine. I know you must have done it well. So, but the idea is, to see if there are people who might have made a mistake or might have judged wrongly, and then we can discuss a little bit. But that's fine. It seems you've understood it well. So, number two, somebody else other than Elizabeth. Please, guys, you just have to speak up. That's it.
The LR plate is the superior part of the fourth ventricle, and of the fourth ventricle gives rise to which of the following structures?
So, the fourth ventricle, what all things it gives? First, tell me that. What are the things that emanate or come out of the fourth ventricle?
Pons and cerebellum?
Yes. Then it talks about inferior, superior, or could be lateral. Lateral is in the third. Remember the thalami, hypothalamus and epithalamus and things like that. So you are coming down in the brain tube or in the brain ventricles. So you're now in the fourth ventricle and you're on the superior side. So there is a thickening which is going on the superior side. What is that?
Is that the cerebellum?
Yes, it is the cerebellum. I mean, if you see the brain, you will realize where the pons is going to form. And also, if you remember the pontine flexure, which is the middle flexure, that is coming towards the inferior side or towards the ventral side, you can say. And that's where the pons is going to be. So on the superior side is always going to be the cerebellum. Okay, number three.
Formation of a unilateral full-thickness cortex in the ventricles is a congenital condition called schizencephaly type 2. Germline mutation in which of the following transcription factor causes this condition?
Yes. So I'm going to ask you quite a bit of clinical and congenital defects. So that's why this question, the first two were more basic. This one is related to a congenital condition. Okay, how many of you know what does NKX2, what does it do in early development? Let's go one by one.
If it's inactive, that's our atrial septal defect, right?
That's correct. So if you have a mutation, there will be an atrial septal defect, and it is one of the pioneering genes involved in cardiogenesis. So it's just a historical perspective. So in Drosophila, when NKX2 was mutated, the flies became heartless, meaning they died, but they are called as heartless flies, which they then called it as tin men. So this gene was called as tin men for a long time. But then later, the Hox genes were classified differently, and so now it's called NKX2. Okay. How many of you know EMX2? And where does it come?
That's the schizencephaly type 2, right?
Yeah, that's correct. And what happens in schizencephaly type 2? What exactly happens? I have written it there, but and how do you do a diagnosis for it?
Well, they have seizures and mental deficiency, and it's where you have a unilateral thickness of the cortex, so there's no like separation.
Yes, so on one side, you do not have ventricles, rather you have the gray matter and white matter invading inside the telencephalon. And you can easily see it if you do a MRI or even a CT scan, either one, right? You can see it as a mass rather than an empty space filled with cerebrospinal fluid. So that's EMX2. And so I think Elizabeth is correct on that. Anybody who knows about FOXC2?
Is that the lymphedema diaskesis?
Yes, that is correct. And there's something associated with it. There are other things that are happening which are happening to, yeah.
Double layer of eyelashes?
Yeah, double layered eyelashes. And that's something which it's a rarity in the sense most people have multiple other disorders, as you mentioned, including lymphedema. But there are people who might have a very low penetrance of this mutation. So how many of you know what is gene penetrance or inheritance, which is superficial, not total? Are you aware of that?
Yes.
Okay, so that's why sometimes people may not see any other, or it will be variable from person to person, and some people are lucky to just get double eyelashes. So that's what FOXC2 is. In any case, let's move on. There are still many more to go. So, okay, number 4. I want volunteers other than Elizabeth.
Which of the following clinical conditions best describes major malformation of the eye associated with congenital galactosemia?
Yes, galactosemia. You all know what is galactosemia?
Yes.
Okay.
It's just to confirm it's an abundance of just galactose in the blood.
Mm-hmm. Yeah. So galactose actually accumulates somewhere and that's what you then observe and you see in which of these out of the 5.
These are the congenital cataracts.
Yes, it is congenital cataracts. Absolutely. So I guess you will be right. Yeah. All right. Number 5.
Division of outflow tract gives rise to which of the following structures?
Yeah.
Is it pulmonary trunk?
That's correct. And which one is the other one?
Trunctus? Wait.
No, not
No, that's not correct.
One side is pulmonary trunk, and then
Aorta?
exactly, it's the aorta because you have that. Remember spiral membrane which is formed?
Spiral septum? Yes.
Yeah. So that's exactly how the OT or the outflow tract is divided. Okay, let's go to number 6. It's very short. The question is short.
The interventricular foramen closes by. Is it A?
No, it is not A. The reason is it initiates the formation of the septum through the ventricular apex, and it is muscular, but then it stalls. It doesn't go all the way up. And that's because you do want to have, in the beginning, some shunting of the blood between right and left ventricle, just like you have it in the atria. And it's only later when the outflow tract is formed, it utilizes this hole to form the aorta on one side, that's on the left ventricle, and the pulmonary trunk on the right side. So which one is correct then? Am I making sense to all of you?
Is it C?
Yes, it is C. So the idea here is that after everything is done, at the end, you will have these endocardial cushions, which are then going to form this membrane to close the intraventricular hole or foramen or whatever we call it. Okay, and that's the end of the shunting in the ventricle and the formation of the two tubes, which is the pulmonary trunk on one side and the aorta on the other. And it's the bulbous cordis which grows so fast there that it closes everything, and including the membrane which comes from the top, right? So you have those triangles, 3 triangles. If you go back and see in the figure, you will know exactly. And I have color coded them, so that's easy for you guys. Okay, let's go to. Okay, so that's correct. Number C is correct.
I have a quick question about the just the wording. Because I know it's like there were like 2 in the lecture. Is interventricular foramen interchangeable with intraventricular foramen?
Yes, that's correct. Did I write interventricular? Yes, it is.
Here it's inter, but like on the slides, it goes between inter and intra for foramen and the septum.
It should be inter because it's between the two. So if I have written intra, then that's wrong, because it can't be there's no membrane formed within a given ventricle, right? So this is correct, inter. Yeah.
Okay, thank you.
Good for clarifying that. Yeah, if I have said it or written intra, it is wrong. There is no membrane in a given ventricle. Okay, somebody else. Number 7. This is easy. This is all about the exciting stuff about stem cells.
I can read it, but I don't know the answer. So, embryonic stem cells need to express which of the 2 following transcription factors in order to maintain its phenotype.
Yes. So you have to maintain. Yes. So you have to maintain pluripotency, right, or totipotency, basically. And so it is. Somebody said A, right?
Yes.
Okay. And you know why? Because it is OCT4 and SOX2. Others, PAX, KLFs are not part of stemness. Okay. Now let's go to number 8. That's a signaling question now.
The intracellular domain of TGF-beta receptor has serine and threonine kinase activity. Upon TGF-beta ligand binding, which of the following downstream proteins are phosphorylated to activate the TGF-beta signaling pathway during development?
Okay, if you've done the TGF-beta signaling pathway, you should immediately know this.
Is the answer D?
And why so?
I believe that the R signifies phosphorylation from my memory.
Does the R symbolize simply receptor?
Yes, it is receptor associated, but also it is not R, but it is R SMAD, which binds directly at the receptor and gets phosphorylated, right? So it's an adapter there. So when you say phosphorylated, there's only one which is phosphorylated, which is SMAD, R SMAD, receptor SMAD. So the paradigm in signaling is that anytime when a ligand binds and activates a receptor, there is a conformational change in the receptor. And in most cases, these receptors have a kinase activity. Here, because it's TGF-beta, you have a serine threonine kinase activity, but there are others where, like EGF, where you can have tyrosine kinase activity, and downstream, immediately you will have a adapter sitting there, which gets phosphorylated, and then the cascade would start. So that's what. Anybody has done signaling in your MSP3 before?
We had to learn some in neuro.
Some in neuro, okay. But those are developmental signaling pathways or some specific to neuro?
They're specific to neuro.
Oh, I see. So probably this is the first time you're getting exposed to all this, yeah.
We did learn about the sonic hedgehog pathway in genetics, but otherwise not the TGF-beta pathway. This is the first time we're seeing this.
Okay, perfect. I'm happy that you're getting introduced, because most major congenital defects you will see is through TGF-beta, because TGF-beta is involved in almost all organs, and including cancer. So that helps. Okay, let's go to number 9.
ANP, atrionatriuretic protein / factor, is produced in the heart by which of the following cell type? I think it should be C, myoendocrine cells.
Yes, that's correct. I think it's a given, in the sense ANP is a hormone, so there is endocrine in that. If you know properly your subject, yeah, you're right. So it's going to be myoendocrine cells, right? 9. So I don't know how many. I think I have more than 10. So yeah, let's see. Okay. Can some volunteer?
During cardiac development, the conus arteriosus originates from which of the following structures?
I believe this is going to be a little tough. Let's see.
Is it D?
That's correct. I think you guys have got it. I don't have to worry at all. So you know there are 3 structures, right? And conus arteriosus is one of them, from the bulbous cordis, right?
Yes.
Yeah. Okay. So you have understood it well. Great. Number 11.
In Rett syndrome, a neurological and developmental disorder associated with inability to use muscles for voluntary movements, which of the below protein that is involved in epigenetic regulation is mutated?
Is that C?
That's correct. It is C. Yeah. How many of you know what actually MECP2 does?
Unlike the other example, like question, it actually puts a cysteine or a methyl directly onto the protein or directly onto the DNA sequence instead of putting it onto a protein.
That's correct. Yes. So it actually recognizes a DNA sequence which has a methylated cytosine. Now, recent published work also show that MECP2 binds to non-methylated cytosines, but then when it binds to non-methylated cytosines, it activates the gene. While if it is bound to methylated cytosines, it actually masks the methylated DNA, where the promoter is mainly methylated, the CPG island as we call it. So you're right. For almost 10 years or 20, not 15 years, I would say, MECP2 was known to be only binding to methylated cytosine, but as the name suggests, but now it is known that it also binds to normal cytosines. But in that case, it activates. All right, guys, I think 11 is more than what we can cover. We have covered almost all in our area. If there is anything I can help you with, you have enough time. I know you have when is your pathology exam?
Monday.
Monday. Okay, so then you have enough time after that. So let me know if there is anything that is confusing or things like that. Let me go back and switch back.
Actually, on the questions for
Sure.
number 11, I was going to ask about D.
11. Question 11.
Yes.
Oh, gee, no, that's wrong. Why the D has been highlighted is what you're saying.
Oh, no, I actually was curious why it wouldn't be D anyways. I wasn't sure if that was the protein things you guys were talking about, or...
Right. No, that's a great question, because MECP2, when it binds there on a methylated DNA, it also recruits HDAC, histone deacetylase, because it not only interacts with a methylated cytosine or a methylated DNA fragment, but by recruiting an HDAC or histone deacetylase, it removes the adjacent acetylated regions so that it completely blocks gene expression. HDACs are for suppression of gene expression, correct? So maybe that was the reason you are asking me about it. Am I correct?
Yeah.
Yeah, no, that's true. But we do not deal with the entire complex which is formed. So MECP2, when it binds to multiple other complexes. So if you read in the literature, it binds to something called a SYN3, SIN3, and SYN3 brings together with it HDACs. So if you see, it's a multi-protein complex which is binding on the methylated cytosines. So absolutely, you're right. It's beyond our scope, so I just put that. But I think if somebody is really reading well in this area, you will come across HDAC and get confused between the two. However, Rett syndrome per se is because of mutation in MECP2 and not due to HDAC. So still, the question holds true, and the answer holds true, meaning it is C. Right? If I had not said Rett syndrome, then it could be HDAC too, because it can bind to methylated cytosine. Okay, anybody with any other questions?
I have one. I'm still kind of confused on how the interventricular foramen closes. I know you had the question in here, and you had the answer for growth of the membrane from the endocardial cushions, but in the lecture you mentioned the bulbus cordis and the ridges. Mm-hmm. So I was just wondering how they're part of this too.
Yeah, so the bulbus cordis comes down, and almost completes the closure of the interventricular foramen, but you do need that membrane to also come down and completely seal the hole. So that's the third component. So if you see bulbous cordis is on either side, right, on that entire structure when it is splitting. And so both of them come down. One goes towards the right ventricle and then gets fused in such a way that the right ventricle now only pumps the blood to the pulmonary trunk. The other part of the bulbous cordis goes and fuses with the left ventricle. Now realize at this point the hole is still there, or the foramen is still there. Only then you can have the left ventricle pumping blood into the aorta. Okay, but you do not want any interventricular interaction or any other blood. So that membrane then descends down and then closes the third part. And so the entire thing now closes, and now you only have a splitting of the pulmonary trunk on one side and the aorta on the other. Is that clear, Lance?
Kinda.
Okay. All right. If you have a question, you email me, okay? So we can go back and forth if there is something.
Okay.
Yeah.
I had a quick question about in lecture 12 when we put, or when in like slide 31, where you put where all the aortic arches, like what they become. Is that like something that we should know in detail? Like the maxillary artery, that one's pretty easy, but like aortic arch number 4 is like the right side, the right subclavian artery, and the aortic arch, like that little table. Should we know that in detail?
To be honest, I have. Tell me which one is it, and I'm sorry. There are so many slides in each of this. What slide you're talking about? And
Slide 31
31
in lecture 12.
Yeah, you do need to know it. The reason is you are also dealing with a very significant clinical or a congenital defect, and you ought to know those. Yes. So it's internal carotid, external carotid is what you're talking about, right? Which are forming what, or where does it go?
Yeah, I remember you being specific about the external carotid, but I was just mentioning the table in general. But yes, I'll know the table. I was just asking if we should know everything in that. I know you did specify where external came from.
Okay. All right. Great.
Thank you very much for the practice questions, Dr. DeVay.
Oh, not a problem. I hope you all do well, and please don't be shy. Again, if you have something, please shoot a question. Okay.
We hope we do well too.
Yes, I'm sure you guys will do well. I think this is a good group, and we have had. To be honest, I'm getting the maximum number of questions from this group compared to previous batches. So I know you all are good. All right. Good luck to everybody.
Thank you.
Mm-hmm.